Okay, so the Enzyme Lab. The Enzyme Lab is one of the labs that I think it's really interesting, but it also tends to confuse people for a number of reasons. Partly there's just the kind of the theoretical underpinnings of it, but then there's also just the mechanics of how we're actually measuring things and how we're actually getting the values for our variables. So I'm going to kind of talk about first kind of the basic idea of the experiment, kind of why it makes sense, why, our hypotheses make sense. We'll be talking about, actually, what you'll be watching some videos of the actual mechanics of doing the lab. So again, I think it's, even though you're not actually going to do it, you're going to actually just use data that from other people. It's useful in interpreting your results to actually see what actually is going on to get the data. And then we're going to spend quite a bit of time talking about how to analyze the data. Because like I said, this is the one that tricks or kind of trips people up, and I'll talk about kind of why it trips people up. So let's just start. Give this Enzyme Lab. So what is our objective? What's the basic question we're asking in this lab?
Does the concentration of hydrogen peroxide substrate have an effect on the rate of reaction with the enzyme catalyst?
Yeah, so basically we're looking at how, at the core, we're looking at an enzymatically catalyzed reaction. And we're asking, how does the substrate concentration affect the rate of the reaction? So if we're thinking about our independent variable, it's just going to be substrate concentration. Our dependent variable is going to be the reaction rate. And obviously we need to have some example of an enzymatically catalyzed reaction to work with. So we're using hydrogen peroxide breaking apart into water and oxygen, and catalyzed by, catalase. So this is our specific thing that we are looking at. Part of the reason why this one is a good one to look at is you can tell that it's going because it makes oxygen bubbles. Some reactions are kind of, we're interested in reaction rate, so we need to have some way to measure that the reaction is going and how fast it's going. So the more that the hydrogen peroxide breaks down, the more bubbles are going to get produced. The faster it's going, the faster the bubbles are getting produced, right? So this is a way for us to have some way to actually get a view at right? Because substrate concentration, this is easy. All you need to do is make a dilution of hydrogen peroxide and you can come up with whatever substrate concentration you want. So, making, kind of playing with the independent variable is just taking hydrogen peroxide and diluting it more or less. In our experiment, we're going to have kind of our stock solution of hydrogen peroxide, which is, it's actually a 3% solution. That's the stuff that you buy in the drugstore. And we're going to have kind of pure hydrogen peroxide. It's not pure, but pure stock solution. On the other side, we'll have just distilled water. And then we'll have a bunch of dilutions, like, 4:1, 4 parts of the hydrogen peroxide to 1 part water, 3:2, 2:3, 4-to-1, and then all the way to this. So, we have these different dilutions from 100% hydrogen peroxide to 100% distilled water, and then a bunch of dilutions in between. So that's going to be the way we quantify our independent variable. We can just look at these different dilutions of the hydrogen peroxide. How concentrated is it? 0 concentration, 100% concentration, or anywhere in between in our different trials that we run. We're going to be running 6 trials in all, right? There's like 1, 2, 3, 4, 5, 6 different times. So we're going to run the same experiment 6 times, each time with a different substrate concentration. And in each case, we want to look at the reaction rate and see how the reaction rate is affected by these differing substrate concentrations. So does that kind of layout make sense? Okay. This part's trickier. How are we going to actually measure reaction rate? I know you've read the procedure, so you have some basic idea.
By the amount.
Say it again.
By the amount of oxygen being released.
So amount of oxygen, but and again, we're not just worried about how much oxygen, we're worried about a rate. Rate is always like something versus time, right? So it's got to be a little more than that.
So how quickly the bubbles start to form?
Yeah, it's like the rate, like how much oxygen is being formed per second, right? That is going to be our measure of rate. Bubbles are telling us that the reaction's going, but how much bubbles per second is giving us the rate, right? If it's going slow, I'm not going to have as much oxygen per second being formed. The reaction is going fast, I'm going to have a lot larger volume of oxygen per second being formed. So my reaction rate is ultimately going to be measured in mils of O2 per second. This is going to be my measure of rate. My reaction rate is going to be measured in milliliters of oxygen formed per second while the reaction is going. And this is a lot less straightforward to get at than dilutions for our independent variable. So, and in fact, I can give you a quick little kind of peek ahead. Basic idea is you're going to have this reaction going in a little flask. Then you're going to have a tube coming out, and that tube is going to go into a burette, and you'll capture the little bubbles coming up, and you'll be able to see how much bubbles have been formed. And you can check at 5 seconds how much oxygen has been made. At 10 seconds, how much oxygen's been made. At 15 seconds, how much oxygen's been made. At 20 seconds, how much oxygen's being made. 25 seconds, how much oxygen's being made. Then we have a slope here. That slope is going to be in this rise over run. This is just going to be mils of oxygen per second. This is going to be our rate. So we're actually going to have to measure the oxygen being produced per second, put it on a graph, get our best fit line, get the slope of this puppy, and that slope is going to be our reaction rate. Right, so to get your dependent variable in here in this experiment, there's actually going to be quite a bit of work involved. You got to get the data to start with, but then you have to graph out the data, and you actually have to do a best fit line, and then you have to do a slope calculation. If you've been in one, like chemistry 114, you do this kind of stuff. This is a classic way to approximate a reaction rate. I think in chemistry 110, they don't do this. So some of you might have seen this method to approximate reaction rates. For a lot of you, it's probably brand new. I will spend more time talking about this in a little while, but I just kind of want to give you a little bit of a heads up. Part of why this experiment tends to be confusing is measuring the dependent variable, the reaction rate, is complicated. And there's more to it than what I've showed you here, because you have to start worrying about, as the reaction kind of poops out, this is not just a nice line. This is actually, it starts going up, but then it's starting to die off. And what part of it is actually the part I care about? And, so we'll talk about this in more detail later. But just kind of putting that out there as we begin, the dependent variable, the reaction rate, is something that you are going to have to calculate after you've graphed the trial and looked at oxygen versus time and found the slope and all that. Okay. So what about hypothesis? We know the question: what is the relationship between the substrate concentration and the reaction rate? What do you expect that relationship to look like? I mean, a lot of people wrote it down, so hopefully you kind of know what you're writing.
I said I predict that the substrate concentration and rate of reaction will have a positive relationship or correlation until the enzyme complex reaches full saturation. At that point, it will level off.
Right, so think about this. We have our enzyme floating around. And we are keeping enzyme constant, right? Every time we do a run of this experiment, we always have the exact same amount of enzyme. So enzyme is not a variable in this. Enzyme is controlled. It's always the exact same amount. And so let us think about, let's put in our H2O2. If I say I put in this much, it's going to get, whenever H2O2 hits an enzyme, it'll break apart at however fast that happens. Now let's say instead of having 2, I had 4 of them in here. I've doubled my concentration of H2O2. There's twice as much for the same volume. Assuming there's enough enzyme, these are going to be banging into enzymes while these are banging into their enzymes. So we're going to be making oxygen twice as fast as we would have otherwise, right? If I just had these two in some certain amount of time, I would have had a certain amount of oxygen being formed. But if I have, these other H2O2s going at the same time with this, I'm going to have twice as much oxygen being made in the same amount of time, right? So it makes sense that if you're increasing the concentration of the substrate, you're going to be increasing the reaction rate. right? Because you're just going to just be more of the reaction happening and more of the product being formed in the same amount of time. So does that make sense? So this is kind of, this should feel kind of intuitive. Assuming that you've got enough enzyme kind of just waiting, if you just keep adding more hydrogen peroxide or increasing the concentration of hydrogen peroxide, you're going to have even more oxygen being made per second because you've got more of the reaction all happening at the same time. So this would be a positive correlation, right? An increasing concentration of substrate, increasing the rate of the reaction. If we're thinking about it in terms of some graph, if this is reaction rate, which we're actually going to be measuring mils of O2 per second, and this is substrate concentration. It could be 0, would just be distilled water. If you have no substrate, there's no reaction, and the more and more substrate, or the higher and higher substrate concentration you've got, the faster and faster this thing is going to go. Right? Does that make sense? Now, the other thing that she was just talking about, though, is this idea of saturation. So we have a limited number of enzymes. Like I said, there's a constant number of enzymes that we're using for every run. At some point, in this 1, 2, 3, 4, 5, 6, 7, 8. Let's say at some point I can add H2O2, and there's still more enzymes waiting in the wings to get that done. But if I finally achieve what's called saturation of the enzyme, where there's so much substrate that every enzyme is working as hard as it can, then something different's going to happen. At this point, I can start adding way more hydrogen peroxide. And it doesn't matter how much more I'm adding, how much higher the concentration is. This dude here has to wait until an enzyme frees up before it gets broken down. So assuming that we've hit this enzyme saturation, meaning all the enzymes are all working as hard as they can, then it doesn't matter how much more hydrogen peroxide I'm adding into the mix. It's still going to just keep going whatever rate it goes when the enzymes are working as hard as they can. Does that make sense? If we were in the classroom, I usually I can I like if let's say I was an enzyme and this was my substrate. So I get here and I get it and I break it apart. And let's say there's a certain rate I can do it. I can tear it at 1 sheet of paper per second. Right? And let's say there's a whole, all of us are here. So there's like 20 people in this, like in Zoom right now. And let's say I was able magically to each, give us pieces of paper. If there were 20 of us all working together and I put 2 sheets of paper in the classroom, 2 people could be tearing at the same time. So we could be tearing 2 sheets per second. We're doubling how fast we're tearing paper. If I put 20 sheets of paper in the classroom and we're all tearing together, we're tearing 20 sheets per second because we're all working together. But once we have 20 people all tearing at the same time, and assuming there's nobody else in the room available to tear the paper, That's the fastest we can tear paper. I could throw 1,000 sheets of paper in the classroom, and we're still going to only be tearing paper at 20 sheets per second because there's only 20 of us tearing paper, and that's how fast we can go when we're all working together as fast as we can. Right? If you saturate the enzyme, every enzyme is working as fast as it can. It doesn't matter how much more hydrogen peroxide is waiting around. The reaction rate is only going to go as fast at some maximum as that the enzymes can do when they're all working together. Is that kind of making sense? So kind of going back to our graph of like substrate concentration. Versus reaction rate. You know, there's going to be some positive correlation until you start hitting saturation. So at some point, you're going to hit enzyme saturation here. At that point, when every enzyme is working as fast as it can, it doesn't matter how much more concentrated the substrate is, you're going to have some Vmax, some maximum rate that the enzymes can do when they're all working as fast as they can. And no matter how much more concentrated the substrate is, the reaction rate is not going to ever speed up anymore. It's just going to be stuck at that maximum reaction rate. So does that make sense? You know, how will we know if we hit enzyme saturation? With our experiment?
Would it just be like a constant stream of oxygen or bubbling?
Well, it's going to be the reaction rate isn't getting faster, even though you are increasing the concentration, right? You can't tell if you are at saturation by looking at any one particular run of the experiment, right? If I have my little flask here, and it's got my little tube going into my thing here, and I have all my little bubbles coming up. I'm measuring my bubbles. There is no way I can know if I'm at saturation or not from looking at this experiment. The only way I know that I'm at saturation is I do the same experiment at, I don't know, at like 40% concentration. What is the rate? I do the same experiment, except now it's at like 60% concentrated substrate. What's the rate? If I've been increasing my substrate, but the rate is not changing, then I know I've saturated the enzyme, that I'm at saturation. But the only way to know you're at saturation is you do the experiment 2 times with different concentrations, but the rate isn't increasing, even though you're increasing the substrate concentration. Does that make sense? There's no way to know that you are at saturation from looking at one run of the experiment. The only way to do it, you know, if I run it, let's say at 10% concentration, I do it at 20%, and now it's faster. I know I'm not at saturation yet because it's going faster as I'm increasing. Then I do it at 30% and it's still going faster. I know I'm still not at saturation yet because I increase the substrate and it's still going faster. And now I'm at 40% and it's like, oh my God, I continue to increase the substrate concentration, but the reaction rate did not keep getting faster. So therefore I know that I'm hitting saturation of the enzyme. Right? It's still bubbles are coming up if I'm looking at it, but the bubbles are coming up at the same mils of oxygen per second as they were at this, and then I go to like, 50%, and it's like, still, it's not going any faster. So I know I'm at saturation. What about just the sheer amount of oxygen? Is this, if I run the experiment with higher concentration of hydrogen peroxide, even if the rate is the same, I am going to have more hydrogen peroxide to break down, right? So what am I going to see different in my experiment if I'm actually running the experiment at 30% and at 50%, and even if I'm at saturation, how are the experiments going to be different when I actually run it? Or how is the run going to look different?
One is going to go for a longer amount of time.
Exactly. I have almost twice as much hydrogen peroxide to break down. So it's going to keep going longer. It's just not going to go faster. And when you're done, you're going to have more oxygen made because you had more hydrogen peroxide to break down in the first place. It's just how long it, in fact, I can show you, if I don't want to, let's actually, I'm going to talk about this other kind of graph later. Let's do this. I don't want to get confusing right now. So whether or not we see enzyme saturation depends a lot on how much enzyme is in the reaction vessel to start with, right? I said there's a constant amount of enzyme in every run. I mean, it's one of the things that if I was actually in the lab, I would have to play with to make sure that the concentration of the enzyme is just right. Like if the enzyme was really low concentration to start with, what would I see as I looked at my like substrate concentration versus reaction rate? Like if the enzyme was just super low to start with,
It would level off faster.
yeah, exactly. I might just like pretty much be at saturation at the beginning and never really see that much interesting going on. What if my enzyme, I have like lots and lots of enzyme, like a really high concentration of enzyme in here? What am I going to see?
It would take a while to reach that saturation point.
I might never reach saturation, even given the concentrations of substrate I'm working with, right? So this actually is a problem in doing this experiment. I have messed it up both ways over the last 25 years, where either people don't see anything or they never see saturation. Ideally, you want to kind of find a sweet spot where there's enough enzymes so you kind of going up in the beginning, but then you hit saturation, and for the highest concentrations that you're using, it levels off so you can see saturation. So this would, you never saturate. Here we hit saturation. Here we're in like always saturation from the beginning, from the get-go. Right, so you won't know exactly what your data is going to reflect until you actually do all the analysis. So you will not know whether or not you reach saturation in this experiment until you do the analysis, do this graph of substrate concentration versus reaction rate, and see what it looks like. Which one of these three options do you get? That would tell you that we never saturated, we were pretty much saturated from the beginning, or we had this kind of linear area where have this relationship, direct direct correlation between substrate concentration and reaction rate, but then we hit saturation where we hit a maximum rate. So any questions about kind of the theoretical underpinnings of this? Yes, no? Okay, so what we're going to do now is go and watch some videos, kind of just showing the mechanics of this. And I should also mention, this is one in order to, I'm pretty sure, hold on, let's double check. Yeah, because I know this is one of these labs that is really challenging often. I spread it out into 2 days for this semester. So we're gonna go over the mechanics and continue talking about it, but on Thursday, we're gonna go even deeper into the analysis and how to think about this lab. So you guys will hopefully feel like, oh, this all makes sense. All right, so I'm gonna go and get into the module. Modules and share. Okay, doing the enzyme lab. And just to kind of give you here, you're not done with this enzyme activity pre-lab. In this background info here on the pre-lab thing, the stuff you haven't done yet, but is down here, videos describing calculating reaction rate, analyzing graphs. This is also going to be interesting for you here. You haven't looked at this yet, but you are going to eventually look at this. And I will talk about this in more detail as well. This is about using, making the graph, calculating the slope to get the moles of oxygen per second. So even though we are now on the next module, you're going to have to go back to this background info because it has info about how to actually get a quantitative value for the reaction rate in moles of oxygen per second. Okay. All right, so the first step is getting the enzyme. And the enzyme, this is a common enzyme in our bodies. Obviously hydrogen peroxide is, very reactive. We use it as a disinfectant, so you want to be able to basically break it down so it's not going to be damaging things. Your liver has a bunch of enzymes to do all sorts of detoxification and this and that. Your liver is the thing that is going to have the catalase enzyme that breaks down the hydrogen peroxide that we're going to use. So this first part here is making the enzyme solution. All right, so lab, we obviously are going to need a source of our enzyme, our catalase, which is going to be in all of our trials to decompose the hydrogen peroxide into oxygen and water. And the source of our catalase enzyme is going to be the liver. So to prepare our enzyme solution, here I've weighed out 15 grams, more or less, of calf liver, fresh calf liver. I'm going to take it and put it in a little blender here. And we're going to add 100 mils of this phosphate buffer. And again, so here, why am I making the solution with phosphate buffer instead of with just water or something?
So that it'll stay at a constant?
So say that one more time.
So that it'll stay at a constant?
What will stay at a constant?
The concentration?
No, concentration is just about volume versus enzyme. What does a phosphate buffer do? Why is it going to be important here?
Resists the pH.
Control for the
change.
pH.
Yeah, it's going to stabilize the pH. Why is that going to be important?
Hydrogen
The
peroxide
pH.
is acidic.
You can
What are-
denature the enzyme if the pH isn't stable and it
Exactly.
could affect the experiment.
Right, exactly. Right. It's like sources of variability. I should have, at this point, well, what are sources of variability? It's like, is our enzyme healthy? Like, is our enzyme getting denatured? Any condition that denatures the enzyme is going to totally mess with the experiment, right? What kind of things can denature enzymes?
Is it temperature,
Temperature, we're going to have to be careful about temperature,
pH?
pH. So we're using a buffer to make sure the pH is stabilized, right? So you need to be careful to stabilize. That would be a huge source of variability. If we were not controlling for pH, that would be a huge source of variability that could affect our results quite dramatically. So, all right, so we'll continue on here. We want to make sure that our enzyme is going to be active, and if an enzyme is a protein, and in order to make sure the enzyme is not going to be destroyed, we have to make sure we don't denature the protein. So you have to think about what kind of things denature proteins would inactivate enzymes. Obviously, one is extremes of pH. This is an enzyme that's been optimized to work in a cow liver, which is somewhere around neutral, around pH 7, 7.4 or something probably. So here we have a pH 7 buffer. So it's going to help make sure it's optimal environment that our enzyme stays stable. We're going to here, so we don't splatter it. Other things that denature enzymes, heat denatures enzymes. So when we blend this, we want to make sure we pulse it more and don't just keep it going and going and going and going and heating it up just from the friction of the blades. Gonna let it think of it. If you have one of those really powerful blenders, you can actually make soup in them. If you just keep it on, it actually gets super hot. So we don't want that to happen here. All right, so now we've made our liver smoothie here. Now we can strain it. I've got some cheesecloth here, and I'm just gonna pour it through here and collect it. Ooey gooey. So here is our little extract of the liver. And obviously this is going to have the enzyme, but I can show you an example so we can test. Does it actually work? The proof would be, let's take some hydrogen peroxide here. Pour some hydrogen peroxide into a beaker. Let's get some of our liver enzyme extract here and add it in, and let's see what happens. Got a good view on that? Here we go. I definitely see the enzyme breaking down hydrogen peroxide as evidenced by the production of the bubbles here. The problem here is the reaction went really fast. Our intention in this is to measure the rate of the reaction, and if this thing just goes off like a bomb like this, we're not going to have a chance to actually measure changes in rate with different substrate concentrations and all. So we're going to have to dilute this quite a bit, like by a factor of 100. So that's the next step here. Is I'm going to take 4 mils of my solution here. If you kind of getting used to this, I just brought this up to the 0, and then I'm going to
just take 4 mils of it. Dispense 4 mils. And I'm gonna just do a major dilution here. This is my phosphate buffer. And I'm just gonna fill this up to about 450. So a little more than 100 to 1 dilution with water. And this might or might not be our ideal dilution. It's kind of hard to tell because every cow's liver is a little different. So we are going to now take our diluted enzyme solution. We're going to check it out with our whole protocol and make sure that the activity is kind of in a range that's fast enough that you're not just sitting, waiting around and getting bored, but not so fast that the reaction happens before you have a chance to measure stuff. So.
Okay. All right, so that is the enzyme that we actually are going to use. We usually actually just mix it up fresh when we do the lab. And you can see it works. It really does make the hydrogen peroxide break down into, you can, and I should maybe, let me get my, the reason why this is a good way to measure the reaction happening, we have H2O2, which is aqueous, becomes H2O, which is also aqueous, plus oxygen, which is a gas. Right? If we are seeing H2O2 turn to H2O, are you going to see anything? No. Water and hydrogen peroxide look the same, and the volume is the same. But a gas, does anybody remember like what 1 mole of an ideal gas is in terms of like volume? At standard temperature and pressure?
Is it 24?
Yeah, it was like 22.4 liters. I think, wasn't that something like that? It's big, right? So I have like a mole of water is, a handful of water. A mole of gas is, 22 liter bottles worth of volume. So you can definitely see the gas showing up because it creates a large volume that is easy to notice as this reaction starts going. And again, we're just going to capture the gas, and we will then be able to see how much volume of gas per second. And as that happens, we can ultimately record it and calculate the reaction rate by our graphical technique that we'll describe later. So let's now go to the next video. Oh, there's a chat. There's a chat. Oh, okay, let's go to share screen. So the setup. This one's going to take 10 minutes. We've got time. This is, again, I think it's important for you guys to have a sense of how are we actually doing this? Because I mean, part of this lab is about kind of learning the mechanics of how do you actually practically do this and what are the gotchas you got to worry about as well. So here we have Tina is going to show how to actually set up that whole system with the little vessel where you can actually do the reaction, capture the bubbles, and be able to measure them.
All right, so what I have set up here is the complete setup for how we're going to do this enzyme lab. So there's the schematics here in your lab manual. And just to go over some of these components, let's take a look at this interesting tube. which is actually just a regular burette. And so most often in chemistry lab, you would have used it this way, where you can open or close it, and then maybe you're doing a titration or something. But in our lab, we're actually going to use it upside down. And so that's what I clamped in here. And I also filled it with water, and we'll show you how the technique for how to do that later. But right now it's full with water, and probably the hardest part of this lab is to read this thing upside down. So whoever is going to be the person that is reading it probably should grab a piece of paper too to help kind of seeing the tick marks. But when the person that's the timer is yelling out now, the person that's reading is going to have to orient themselves and see where is the level of water. And so, of course, now 50 is up here and 0 is down there. And so, for example, if the level was here, that's actually 47.5. And then this would be 48, and then so forth. So just take a moment and get really comfortable reading this, because it is a little bit stressful when you're going through the experiment. But keep in mind, each trial is super fast. If you do mess up, you can just redo it again. So back to our setup. So what we have here is a water bath. We filled it kind of halfway up with just plain tap water. And in this case, I have filled this one, and we're going to zero that later. But before that, we're going to take a look at some of the other equipment. And so what we have here is just a Erlenmeyer Flask with a stopper. It has 2 glass tubes and 2 hoses here. The shorter hose you're going to use for your syringe. And so this syringe has milliliter marks on it, and this is where you're going to do your dilution according to this table down here. So each trial is going down a column, and it tells you how much water and how much hydrogen peroxide you need. And so when you're doing this, it's essential that you start with the water first, and then move the syringe over to the hydrogen peroxide, just so you're not cross-contaminating and messing with the dilution or the concentration. The other thing that I think is pretty clear, if you just take a look at these 2 solutions, they both look like water, which means that you need to be super organized and label your containers. Even with these bottles, it can happen that you go over to the counter and pick them up, and a student in the previous section might have put the wrong cap on, or, it's just not the right thing. So being super organized, keep it labeled. You can use the syringe directly into this. However, when it starts getting low, it's hard to get in. If you do pour it over into a beaker, then of course, label your beaker. So it's easy for everyone in your group to see which fluid is what, and that makes it easier for that. So then after you make your dilution, you're going to stick the syringe on the shorter tube. Someone else in your group has probably been nice enough to go over and get the enzyme extract that we made earlier. And you're going to have the same amount of enzyme for each trial. So it's always going to be 10 mL, which means that in this experiment, we're keeping the catalase concentration constant. What we're varying is obviously the concentration of hydrogen peroxide for each of the trials in the experiment. So once we're done with that, we're going to seal this tightly. You have your trial dilution ready made in this. And then once you squirt the hydrogen peroxide down to the catalase will help out with that reaction that is going to lead to water and oxygen. And the oxygen is a gas, which is of course going to want to escape and bubble out of this tube. So what we're going to do is we're going to immerse this before we start the experiment and stick it up into the burette. And then it's kind of key that you also make sure that you don't have it accidentally slip out during the experiment. But if it does, remember, this is a super fast experiment. You can just redo it, the trial. So during the experiment, one person is going to agitate this. We keep the reaction going and everything is mixed nicely. So then gases are going to form, or oxygen is going to form and start bubbling here, displacing the water. And then the person that's reading the graduated cylinder will yell out the numbers, and another person will record it into the lab book. So it's key that you have made a table so it's quick to record times and volumes as it goes along, because things will happen quickly. And then new for this time, we haven't done this before, but we have experienced sometimes that we get enormous bubbles traveling up, doing a major displacement. And so this time we're gonna actually try putting some of this gauze or cheesecloth that has large holes in it. We're gonna try to break up those big bubbles by putting this on like this and insert that into a burette, and hopefully get a little bit more finer bubbles, so it makes it easier to read the various readings.
All right, so I'm going to show how you set things up for the beginning of each trial. To start, we have already, my lab mate has placed 10 mils of our diluted enzyme solution here in our little Erlenmeyer flask. Make sure before each trial you actually kind of clean this off, do like a rinse with distilled water, so you have a kind of clean, fresh enzyme each time. And then I'm going to need to have my appropriate dilution of my substrate, which I'm going to put in my little syringe here. To illustrate, I'll show, for instance, trial 2. Trial 2 is going to be 2 mils of hydrogen peroxide, 3 mils of the distilled water. In each case, we make sure.
And just, right, this is what I was talking about when we were talking about independent variable. These are just, each of these trials is that dilution of hydrogen peroxide, either pure hydrogen peroxide solution, which would be like 5 mils H2O2 and no water, or pure water and no hydrogen peroxide. And then we have the whole range in between. So that will be our independent variable is. And notice, why are we kind of doing it this way? Like with the numbers, like, why do they always add up to 5? Why is that important? Anybody?
Maybe so we're keeping the amount of solution the same, but we're just changing the concentration.
Exactly, right? Right, we have our little flask here. We have like the enzyme. We have the H2O2 solution. And then this is our reaction. Another thing that can, if you think about what kind of things affect reaction rate, the total volume that this is occurring in is going to make a difference, right? So we want to keep the volume constant. So so the volume is another thing that we're making sure doesn't change. The only thing that's changing is the concentration of the substrate, but the total volume of our reaction is staying identical each time. So again, as much as possible, when you're doing an experiment, you want to kind of vary the IV and keep everything else constant. We're keeping the pH constant, we're keeping the volume constant. So the hope is, if the only thing you're changing is the independent variable, then if you see a change in the dependent variable, then you can say, responsibly, like, oh, that's due to my change in my independent variable. Whereas if you have other moving parts, then maybe it was my independent variable, but maybe it was that other stuff that I didn't control so well. Damn, I can't really interpret my data very easily anymore. So just kind of putting that out there. All right, so let's continue with this.
There's a constant volume. Again, another source of variability in this reaction is the total volume that the reaction is taking place in. So we want to make sure that each trial is the same volume so as you heard, we want to make sure we start by bringing up our water. So I'm going to look here. I need 3 mils of water. All right, so I am going to suck up my water here. Again, I need 3 mils. So I actually pour in a little, suck in a little more than 3 mils. And then you can knock out all the air bubbles. And you bring the plunger up until the black of the plunger is exactly on 3 of the marking. That means there's 3 mils that are going to get squirted out of water. And then for the hydrogen peroxide, the 2 mils, we just have to dip the nose into the hydrogen peroxide here and continue sucking up until I get exactly to 5 mils on my marking. And there I go. So there is 5 mils of liquid in here, 2 of the hydrogen peroxide, 3 of the water. So that's my dilution for trial 2. This then gets very firmly attached onto the short tube of this little setup going into the stopper. You really want to make sure that this is sealed well and isn't going to fall off when you're going through the experiment. Here we have our 10 mils of the enzyme solution, which is constant from trial to trial. I also want to make sure I have a really good seal between my rubber stopper and the flask. So I'll use some water on the stopper before I put it in. Then I can put the stopper in and really seat it very, very firmly. Obviously, don't push it so you're going to break anything, but you really want to have it firmly in there. And even though it's pretty darn low, you will be able to kind of rock it and get it out when you're done to do the next trial. So now we're kind of set here. When I want the experiment to start, I just squirt the substrate into the enzyme. The reaction will happen. Any gas, oxygen that gets produced will exit through this hose. So this hose now is going to have to be placed in the burette where I can actually capture and measure the amount of oxygen that's getting created. So I'm going to basically reach under here and put it here. I want to make sure it goes far enough up that it doesn't fall out as we do the actual experiment. And so what we're going to do in a few moments is show you the actual experiment in progress.
Okay. All right, so you all kind of get a sense of the actual physical setup. You know, it's relatively crude, but it actually does work. And so the last part, we might as well just do this. Is 7 minutes. Just show you what one run actually looks like, you know, and then we'll like wrap up a little bit here, and I'll talk a bit. I think we'll wait until next class we'll spend more time looking more in detail about how to, like, what we find and how we analyze it. But I want to kind of finish up the basic mechanics of actually getting the data, like what our question is, how we're actually doing it, and then we'll spend more time in this as the week goes on, like how do you actually kind of analyze it. But let's take a look here. Performing a trial.
Okay, so the first thing we need to do is fill the burette with water. And just so you don't make a big wet mess on the floor or someone's pants, make sure the stopcock is turned this way.
Sorry, we can't see your screen.
Yeah, we can't see it.
You can do this over at the sink, but
Yeah, oh, that's funny. All right, let me try that again. Sorry about that. All right.
Okay, so the first thing we need to do is fill the burette with water. And just so you don't make a big wet mess on the floor or someone's pants, make sure the stopcock is turned this way. That way nothing will run out. You can do this over at the sink, but maybe even smarter is to just grab a little beaker here. And if you are steady on your hand, you can just pour it in. You want to fill it all the way, not just to 50, all the way up. Stick your thumb on the end, invert it, and then make sure you aim so the numbers are easy to read. So once you're under the water, you can let go of your thumb, and we're just going to clamp it. Like that.
Right, it stays up, kind of like when everybody's done that, where you put your thumb on the top of a straw and you can lift the water up, right? So as long as the top is sealed, it creates a vacuum where the column of water stays up the tube there.
That will be a good position. Also, so we don't need to stretch the tube. You can scoot the bath a little bit closer. All right, so we're ready to start our trial tube test experiment.
All right, so again, I have my little reaction flask ready to go. I just need to make sure that I have this tube now up in the burette so it'll capture the oxygen. So I'm gonna take a little piece of gauze here. Again, we're gonna see how this works to try to diffuse the bubbles a little and break them up. I'm gonna take this and I'm gonna push it up into the tube. Now you want to make sure you go up, at least like maybe a couple of inches, because you don't want the tube to fall out of or get pulled out of the burette as you're going. Because again, if it does happen, you just have to redo the trial. And it does happen every once in a while, but you try to make sure it's up high enough so that won't be an issue.
All right.
It also means when you're mixing and agitating, you gotta kind of stay close. Be careful, if you get too excited, you'll yank the tube out and have to start again.
All right, so before we start, we also need to zero the water level now at 50. And so we fill the whole burette. So this is, you just gently kind of twist it, and then you're gonna get the water level to go down slowly. There.
And this is not quite always as easy as she makes it look. There's kind of this delicate sweet spot, but once you have practice, you'll be able to do that.
All right, so if you go lower, then refill the burette and set it up again. All right, so we're ready to go now. So David is gonna inject our hydrogen peroxide into our chamber, and I'm gonna be reading the volume every 5 seconds.
And again, we typically have 4 people working at this at the same time. There'll be like the timer person who is going to do the time, and every 5 seconds. And you could just say mark, time, now, whatever. It's just every 5 seconds, there is a time mark. There's gonna be the reader who is actually reading what is the volume of water at the moment they heard the yell time, which can be a little flustery because there might be bubbles coming up and it can get somebody on your team who's got nerves of steel. And you'll also have kind of the reaction master person that'll be injecting, mixing to the substrate and the enzyme and keeping it agitated for the whole duration. And then there'll be the person recording the data. It makes sense to usually, I don't know if hopefully this focuses, some kind of a chart ahead of time. And this is already, trial 1's already been here. But here I know the time is going to be in intervals of 5 seconds. I've got a space here for my burette reading. And then what I'm really interested in is the oxygen, which is going to be 50 minus whatever the burette reading is. So I kind of have that set up. So it's going to be nice and easy and clear to
right? Because the burette is going to start at 50 mils of water. And for every mil of oxygen that goes up, it's going to push the water down a little bit. So the thing you're measuring is the mils of water, but you're going to have to do that simple math. If there's 50 mils of water, there's 0 oxygen. If there's only 45 mils of water, that means 5 mils of oxygen have gotten in there to displace it. So that's something you're going to have to actually do. Like the data we're going to give you is going to be the water level. So you're going to have to do, the first step will be to convert that into mils of oxygen, which is just 50 minus whatever the water is.
Calculate in my lab notebook. So normally we'd have somebody recording that well. So here we go. All right, so we are good to go. I've got my timer ready to start. I've got my reaction vessel with my substrate and my enzyme. And I'm going to hit start and make sure I get all of it in here and read.
49.5.
Read.
46.3.
Read.
42.2.
Read.
33.1.
Read.
29.2.
Read.
26.4.
Read.
25.0.
Read.
24.1.
Read.
23.0.
Read.
22.7.
Read.
22.4.
Read.
22.1.
Read.
22.0.
Read.
22.0.
Read.
21.9.
Read.
29.1.
Read.
29.1.
Read.
29.1.
So that is actually, normally if you get the same reading 3 times in a row, that means the reaction has run its course. So there's no reason to keep taking data. We also kind of hit 90 seconds there, which is also typically what we're interested in is when the reaction is really rolling along. And in all the cases we're going to be doing, 1.5 minutes in, you're on the tail end of the reaction anyway, even if there is still a little bit of oxygen being produced. So usually at 90 seconds, you can call it and move on to the next trial. But that was awesome.
Okay. Yeah, and she makes it look easy there, but it really can be tricky trying to read those numbers off as the bubbles are coming up, and it's 5 second comes up so fast. Typically, we end up having groups get started where people totally and then have to just set it all up and start again. So what I want to do is I'm going to spend a couple of moments giving you a sense of where where you go next with this and where some of the confusion arises. So basically, you are going to end up getting a graph for each trial. So for any particular trial, there's going to be time in seconds and 5, 10, again, 15, 20, 25, 30, 35, 40, 45, 50, right? You're taking readings every 5 seconds. And then you're going to have mils of oxygen, which you're going to get, that's going to be 50 minus whatever the burette reading is. Then you're going to look at this, and typically it's going to look something like this. Typically, it's never going to be beautiful, but in general, it's going to be something where it's kind of going up and then kind of dies and then kind of gets flat. So this is a typical trial. And what is happening here as it's flattening out?
Enzyme saturation.
So that is not, this is where people get confused. So this is not having anything to do with enzyme saturation, because remember what I was mentioning, enzyme saturation, you have to look at another graph that looks superficially similar. If we look at mils of oxygen per second, aka reaction rate, and substrate concentration, and I see as substrate concentration gets higher and higher, there's the saturation, because I'm increasing substrate concentration, but the reaction rate isn't getting any faster. It looks superficially kind of like this shape, but it's radically different what you're looking at. This is looking at the time versus the volume of oxygen in one trial, right? There's no way you can tell if we're in saturation in one trial. You have to do multiple trials and see like that the reaction rate isn't getting faster as you increase the concentration of the substrate. So kind of rethink this in terms of it's a single trial, time is going on, and the volume of oxygen isn't changing anymore. So what is happening here?
Could it be the enzymes already broke down the hydrogen?
Exactly.
The water and oxygen?
Yeah, so does everybody hear a motto there? Right? The reaction's over. Basically, on this part here, the hydrogen peroxide is being broken apart by the enzyme. We're making bubbles. At some point, you've run out of hydrogen peroxide. It's all broken down. The enzyme is sitting around, but there's no more hydrogen peroxide. It's all broken down into water and oxygen. So you can wait till the cows come home and there's not going to be any more change in oxygen level because there's no more hydrogen peroxide to break down. So this is nothing to do with saturation. This is reaction is done. So, but again, this is where people get confused because it looks really similar in terms of the shape. And in fact, if I'm actually interested in the reaction rate, I don't want to look here. This area here is boring because the reaction is dead. That's not when the reaction rate is going to be measured. I want to measure the reaction rate down here. So this is where I'm going to actually take my slope. Calculate reaction rate. This is where reaction is going strong. So this is important when you are actually, and again, I will go deeper into this on Thursday, but when you are looking at your data, you are going to have to make a chart like this. You are going to have to figure out, nah, this is not good. This is the part I care about. Do a best fit line, calculate the slope, rise over run. That is going to be, this is going to be the slope, is going to be the rate. And then you're going to have to go onto this graph, and for any particular point, that is gonna be that rate. For whatever trial that was, whatever substrate concentration you're using for that trial, you calculate that rate, there's the dot. You'll do some other trial and calculate some other rate, and that will be that dot. But to make this graph, you are gonna have to make like 6 of these graphs. Does that make sense? Yes, no?
Yes.
Okay, so next time we'll look deeper at the data. So yeah, so today was more about kind of setting things up, both in terms of hypotheses, objectives, and mechanics. And then on Thursday, we'll look much more deeply into the actual analysis of the data. How do you actually get your dependent variables calculated and actually check to see if the hypothesis is supported and all that. So, and it's 12:27. So I'll still be here for office hours, but if in terms of our class, we are officially done. And